So my name is Mathieu Petitjean. I am the Chief Engineering Officer and founder of PharmaNest. And PharmaNest is a company that does digital pathology and AI out of Princeton in New Jersey. Oh, yeah. Well, that's a good question. So I am a pianist, so I qualify into the 3M phenotypes, which is the music, math, and medicine. Mm-hmm. And I also like to ride Harley Davidsons in New Jersey. Yeah. No. I'm trained as an engineer and solid states physics, so I'm more into the mathematical aspects. Yeah. So no, unfortunately, I don't have a favorite molecule. So in our digital pathology and AI paradigm, we wanted to quantify the phenotypes of histological fibrosis, right? Mm-hmm. And pathologists have told us that, fibrosis looks different according to etiologies, disease, therapeutic response. So we developed this platform that basically goes down to every single fibrosis fiber in a histological image, quantifies that in multiple dimensions, and with those data sets, we can have insights to the phenotype. What has really changed the way we collaborate with biologists, because there was a gap between, okay, what does this data mean and the biology? So what has closed the gap is when we introduced a year or so ago, the fibromaps. So take single cell analysis where you have a cell that is quantified in multiple quantitative dimensions. And single cell analysis has revolutionized the biology and the study of biology. Biologists understand that. So we have introduced single fiber UMaps and a tool that we call FibroMap that really uses several clustering methods to give some sense to different fibrotypes that exist in a tissue. And that has really helped us connect between our mathematical and AI algorithms and the brain of pathologists. So I don't think it's a major technological breakthrough, but I think for us, it has connected the brains, and we expect a lot. In fact, we think that single fiber analysis is going to revolutionize the study of fibrosis in the same way as single cell analysis has revolutionized biology. We are a small company, so we are actually very, very focused. So we do the single cell analysis, and we have computational methods that run on the cloud to really enable single fiber analysis. FibroMaps now link these huge data sets with biology-friendly frameworks. And I think where the future is taking us, and basically answers the question, is when we overlay our single fiber spatial data sets with single cell spatial data sets, right? So we published that this year in August. And what that does is basically explain what's the biological significance of a specific fiber phenotype. And the second thing that it does is that it creates spatial data sets that combine the molecular view and the histological view and provides insights that can be summarized into novel biomarkers. Right? So I think this is where we're going, and we're going to see these things coming up in the coming 12 to 18 months. Oh, that's a difficult question. We are not kind of an ECM company because we chose to simplify ECM and really focus on histological fibrosis. Mm-hmm. But that might be the answer to your question is, it's extremely complex. Over these past days, we've been from, atomic force microscopes to X-rays, multiple orders of magnitudes. It's highly complex. So I think successful companies will be the ones that perhaps oversimplify the matrix or focus on something and really stay focused on it for several years. Number one, become very, very good at that specific dimension of the matrix. And the other thing, that's find a way to monetize or to really explain the value before monetizing of their findings in a very complex environment. So that's probably what's going to make companies successful in the space. I think there are twofold. First, curiosity. I think we are very, very curious. In prior life, I worked on, pattern-enhanced, stiffness-enhanced cell behavior. So that was fascinating for an engineer, always to see cells behave differently when you put them on the pattern or when you don't. So that's curiosity on one end. And the other end is basically the community. We've been working with the community for the past 5 years, and they thought it would be interesting to have me participate and explain what we do. And this has been extremely rich over the past 2 days in terms of interactions and discussions, and say, haha, I've been looking for this forever, so can you help? So I think this is why we connected. During my Ph.D., I had the opportunity to work with a lot of very strong mentors. Some of them were recognized with the Nobel Prize. But all of them told me to trust my intuitions. So I think this has helped me a lot, in particular in fields where there is not a lot of established science, right? And trusting one's intuition has probably been the most rewarding advice that I got over the past years, right? So I think if there are young scientists listening to this interview, I'd probably cascade that to them. You've been trained, you've met a lot of great scientists and mentors, but trust your intuitions, take a risk, and I think it will work. The first is scientifically overwhelming. I think it's related to the complexity of the matrix, the quality of the speakers. So really, really overwhelming and humbling at the same time. But I think the other aspect that was wonderful is that we come from totally different angles, different views of the matrix, and then we connect and say, okay, there's a way to collaborate and build things together. So that's perhaps the two takeaways that I have with me today. Dogs. Collagen, definitely. Collagens are the plural.
That's correct. Thank you so much.